generalizable, um, how do we say something generalizable across a population of people at the level of studying single neurons in that way, right? The same neuron doesn't exist in two different people's heads. And so as we try to figure out how we can bridge this divide, we know that at the limit of the fidelity of our techniques, the whole thing is going to fall apart. On top of that, the more that we study it, the more highly specific it seems, right? Each individual pattern of activation, each individual multivariate analysis, right, reveals something about the brain response, but it also reveals that in different contexts, that response is very different. In addition, we can't assume things like stationarity. We know from a wide variety of studies, including one of which I'm going to try to get to today, that behavior is not stable across our independent trials. And so we're not dualists, so therefore the underlying physiology must also be changing. And so how do we deal with this system that at the limit is going to be different, right, across different people's heads? And which is constantly changing and adapting itself in response to our queries. And the thing that I've settled on really is trying to look for those aspects of the underlying physiology, right, which will carry through into the behavior, right? In essence, studying not exactly the flow of the river, right, but its banks, right? What are the constraints that are going to be present even in the high-level descriptions of the behavior? And in doing this, I kind of turn a little bit on its head some of the sort of standard ways that we go about inquiry, right? The way that we oftentimes work is we have a well-described cognitive model, right? Here's Baddeley's, one of Baddeley's later figures of his model of working memory. We then design a paradigm that's going to investigate some specific aspect, right, of this model. Then we look for activation, right? And we get some pattern of activity, and we point at some part of it and we say, here, this is the part that's supporting, working memory or supporting working memory maintenance or whatever it is that our paradigm was designed to get at. In fact, that's proved to be really difficult to do, right? The answers that we come to oftentimes differ. And so what I'm going to try to do in this talk is to work the problem sort of backwards. So not to throw away our observations, right, that come out of fMRI and so forth, because I think they're very important, but rather than just trying to kind of directly relate them to our cognitive models, instead to take a detailed look at what we know about the physiology, about the response of the regions that we observe, and see whether or not we can use the details of that physiology, right, be it knowledge about cellular molecular bases or tuning curves or multivariate patterns of response, and then make some particular set of predictions about behavior, right? The idea being to try to take the physiology and use it as a basis for making a novel set of hypotheses, right? Ones that we would not have come to before, and seeing whether or not those hypotheses actually play out, right? Which gives us greater confidence that the physiology we've observed is really relevant and begins to kind of try to bridge this divide. And at the extreme, these sorts of observations and these tests of hypotheses can also help us to refine our ideas of the original cognitive model. And so moving away from just trying to situate our models within the brain to trying to have a more bidirectional interplay between the models and the brain data, and trying to get them both kind of into alignment, right? Trying to make our models more biologically plausible. I'm going to try to play this game in a bunch of different domains today, which are sort of domains that I've touched on over the course of my career. So we're going to start off looking at two sets of domain-specific mechanisms, right? These are particular observations of the way in which the brain is sort of functionally organized. And what we're going to do is try to relate that functional organization, in the first case, to large-scale neuroanatomy derived from homologous work done in the cat. In the second case, we're going to look at the consequences of the co-localization of working memory maintenance and perception. And then finally, we're going to get into some domain general mechanisms where I'm going to try to take general known characteristics, right, of how neurons adapt themselves, how they learn, and see whether or not those characteristics can be found, right, in behavioral data. And so there we're going to do both laboratory experiments and then also a novel set of big data gleaned from about 15 million participants. But we're going to have to go quickly. So forgive me if I kind of give short shrift to anything. So to begin with our category selectivity, it's helpful to think to take a brief look historically at what defines our notion of the broader circuit in which these category selective regions sit. So the original sort of and currently still really dominant characterization of vision is at least at the cortical level, a bifurcation where information enters early visual cortex, and then some of it is going to drive responses down the inferior temporal lobe that are going to be critical for things like identification and object recognition, and then some is going to drive responses into the parietal that's going to be critical for vision for action, for localization. And the original studies that demonstrated this dissociation were really monkey lesion work, showing that monkeys could or could not learn tasks on the basis of spatial or non-spatial factors, depending on the lesions that were applied. This leads to a very strong and elegant sort of inductive account from the neuroscience scientific data of vision, right? Which is you take the sort of basic observations of Hubel and Wiesel about hierarchical processing, and then you try to play that out, right, over a larger circuit. And so at this point, we think of the ventral pathway as sort of a series of visual areas, right? Each one of which is going to respond later, right, as information works its way through, and then each one of which is going to have an increasingly complex set of representations within them. And this basic idea really is what underlies even the sort of modern computer vision algorithms like the DNNs. All they're doing is playing this same game out, right, over a much larger model than we had looked at previously. But we run into a problem, which is once we leave the domain of sort of early to low intermediate level vision, and we look at the human fMRI data, what we see is something quite surprising, right? Which is a bunch of category selective regions. So these are areas that show a response that is selective, right, greater for one category than for others. And moreover, not only do we observe these regions, we observe a number of them, and they also occur in stereotypical locations, right? And at first glance, there's not really a good way to square this, right, with the hierarchical processing idea, right? There's nothing in that hierarchical model that is going to directly produce stereotypically located category selectivity. It may produce occasionally clusters, but those clusters are not going to end up being in the same place across different instantiations of the model. And so that led some to propose that category selectivity in the cortex is arbitrarily placed, right? It's the result of a bunch of, at the extreme, sort of mutation events, which are going to co-opt sections of cortex for dealing with these biologically relevant, highly important categories of stimuli. And so this is a figure stolen from a paper by Edward DeHaan and Alan Cowie, where they basically proposed exactly this, that once we move beyond striate cortex, in the human at least, what we're dealing with are these loosely connected modules, such that modules which share selectivity are more densely connected, and then only weakly connected to modules with different kinds of selectivity. That idea is an effective description of the fMRI data, but it produces a real problem, right? Which is now the relationship between early and low intermediate level vision and category selectivity is totally arbitrary, right? In effect, even though we're studying what we think is an integrated circuit of neurons, we have effectively placed a dividing line between them. It also opens up an infinite quest for new areas, right? If in fact we think of sort of extrastriate IT cortex as being tessellated with these areas, then it's perfectly reasonable to go out and look for the next one and the next one after that. And there's not really a good way of curating that, right? And so what we want to do ideally is find a way to get these two things to actually agree. And the argument that I'm going to make is that we can do that, right, by looking in detail at the gross scale connectivity between regions. The first thing that we have to do, right, is acknowledge that this account of an arbitrary organization is almost certainly not right. And the reason for that is, to my mind, most clearly demonstrated by the visual word form area. Right, this is an area that has all the same hallmarks of category selectivity as all the other areas that I've shown already for faces and bodies and so on, right? But it's now selective for orthography, for the visual word form. There's an associated disorder that results from lesions in and around this area of the brain, but it cannot possibly be the result of evolutionary pressure. At least in the United States, literacy in the general population is, being kind, maybe 200 years old. It's not enough time for any particular event to have occurred to have created this area. Instead, there must be some other constraint which is capable of producing stereotypically located category selectivity. And so that was the impetus for this project that I engaged in over a couple of papers with all the collaborators that you can see at the bottom, where what we tried to do was to update the characterization of the ventral and dorsal pathways with about 30 years of macaque tracer work that had been done since they were originally described. And so throughout the talk, I'll have pictures of my collaborators, without whom obviously this work is completely impossible. I'll take a moment here to just call out the character on the far left, who is Salim Khatar-Bacha. Salim is absolutely encyclopedic in his knowledge of macaque neuroanatomy. And so what he did was to pull together all of that knowledge into a single sort of description of the actual neuroanatomy of the ventral visual pathway, which is what you see here. I don't want you to kind of try to get into the details of all of it. Suffice it to say, it is not simply a staged hierarchy that works its way down the inferior temporal lobe. There are a number of highly specific connections. There are a number of connections that skip intermediate levels. And in fact, there's enough detail, right, in exactly how these connections are set up that I believe it can be used to explain category selectivity. Nor is it the case that the ventral visual pathway is somehow really just dead ending in the anterior temporal cortex. It also has a set of really complex projections. And again, this is in the macaque, and assumedly the human connectivity is a superset of this, if anything. It has a dense set of interconnections with the hippocampus, with the orbitofrontal, with the ventrolateral prefrontal cortex as well, all of which are going to act to constrain the sorts of representations that are actually going to be formed here. And so when we think about our computational models, things like the DNNs, what's important to remember is that those are models of a computational principle, right? What can hierarchical processing produce? They are not, however, models of the structure of the brain. So when we begin to apply some of the constraints that come from the neuroanatomy, we can make pretty firm and, in fact, borne out predictions about where category selectivity is going to end up being. And so the general argument I'm going to make, and then I'll show you a specific study where we tested it, is that the reason you get stereotypical selectivity is because the neuroanatomy is itself stereotypical at the level of the gross connectivity. There is simply a fundamental problem of packing this many neurons into a skull, and that's going to mean that certain areas have access to information and other areas do not. And so depending on where the information needed to drive an adaptive behavior is, right, that is where experience is ultimately going to cause the formation of a category selective cluster. Okay, so we're going to test this idea out in the domain of scenes. And scenes are great because they are so complex as a visual stimulus that when you say that a region represents scenes, it's actually highly ambiguous as to what that means, right? So inside any particular scene, right, you have different objects, right? You have different scene categories. This is a hotel room. You have different geometry, right? How navigable is the room? Where is the room situated in space? What's the actual size of things? How far away are items, right? These are all things that we effortlessly deal with, right, in the visual world constantly, and all of which could form the basis of a scene representation, right? In addition, scenes are extremely different from one another. And that provides us with the opportunity to really run, as we'll see in a moment, a data-driven experiment, trying to figure out exactly what the nature of scene representation actually is. Okay, so what are we going to do? Well, first we need a target area. So we're going to take the parahippocampal place area, which is this highly scene selective region. It's been described now since 1998. Here you can see it in a schematic. Over here on the right, these are actual slices of 7T data. And what we're going to do is try to predict which aspect of scenes is represented in the PPA based on where it sits in these broader circuits. So we look into the macaque data, we find the homologous area, not surprisingly the parahippocampal gyrus. In the macaque, it contains these two areas, TFO and TFTH, which receive a hugely strong set of inputs from the ventral portion of the V4 visual field map. And moreover, the projections that come out of V4V into TFO, TFTH are primarily coming from the peripheral portions of that visual field map, right? As opposed to the more foveal bias that we see in TEO. Right? We can see some evidence for this in the human as well. This is a figure from, I think, one of Rafi Malik's classic studies, where he's just looking at the center-periphery biases, and he shows that on the medial inferior surface of the temporal lobe, you have a bias towards the peripheral portions of space. We can also look at the descending connectivity coming into this area. So here is a pathway emerging from the posterior parietal cortex in the macaque that's providing descending spatial information coming from the parietal into TFO, TFTH. We can also see the dense interconnections between these regions and the retrosplenial cortex and the hippocampus. So on the basis of, and there is some evidence for a homologous pathway in human functional connectivity data. This is a slide from one of Margulis's studies. And so what we're looking at here then, right, is a region that should be heavily biased towards the spatial aspects of scenes. It's primarily sensitive to peripheral visual information, and it's receiving descending input from the parietal. But we want to run an experiment that can test that without introducing any bias into the experiment. So here we lean on the natural variability available to us in scenes and simply present as wide a set of scenes as possible now to human observers in an fMRI experiment. So the scenes, we obviously still have to make some choices, right? We can only present at this time 96. And so we needed to kind of select them from a broad array of different categories, different geometries, whether you're in an indoor or an outdoor space, enclosed or not, man-made or natural, so forth and so on. But importantly, what we're going to do is present each one of the scenes individually and simply observe the multivariate response of the PPA and then see how similarly or differently it treats all of these scenes without imposing any particular structure. Okay, because we're going to present each scene individually, that allows us to treat each scene as a condition. What we can then do is take that multivariate response, and here I'm just going to summarize it in a multidimensional scaling plot, right, where the distance between any two pairs of scenes is going to be reflective of how similarly it's treated by the PPA. Some of you can already see the structure which is emerging from this. It's a little bit easier if we make it symbolic. So here's the same configuration, but now instead of scenes, it's just going to be dots. And you can see here all the man-made and natural scenes are really intermingled. There's not much of a difference. Same basic story for scene category as well. Here is near versus far. This is how much depth there is in the scene, if it's close up to you or if it's farther away. And you can see a bit of a separation where maybe most of the purple is sort of ending up towards the bottom. But when we look at this distinction, whether you're in an enclosed space or not, you can really see, right, that the response of the PPA effectively groups all the open scenes on one side and all the closed scenes on the other. Okay, it's always nice to have a control region to make sure that your effects aren't too general. This is early visual cortex, area analogous to V2 through V4, the peripheral portions of it, and you can see it really doesn't honor that difference at all. What we're going to do here is our first pivot where we try to make a behavioral prediction, right? So if in fact this region is capturing this aspect of scenes, then we should be able to see the microstructure of its representation apparent in behavior. And so what we did was to, in a separate group of subjects, arrange a series of matches where we would present two scenes and simply ask the participants to choose which one of them was more open or closed, or more man-made or natural, or so on. And then across all of these different matches, we can derive an ELO rating using the same map that they use for chess players, and derive behaviorally which scenes are sort of the most open or the most closed. Okay, and then we can directly compare that with a metric derived from the physiological data and expect to observe a correlation between the two, which is exactly what we find. So this is suggestive, right, that the individual variation in the physiological response to scenes in the PPA maps onto a behavioral decision. Okay, and the same relationship obviously did not hold for the early visual cortex. Okay, but we can go further than this, right? There's another mysterious aspect to these regions beyond the fact that they exist, right? It's also the fact that there are two of them, right? Where you have one area on the lateral surface of the brain and then one area on the ventral surface. And there's not really a great explanation for this. On the face of it, it appears to be quite redundant. There have been attempts to demonstrate that these areas are hierarchically organized, right? With the lateral surface providing input, right, to the ventral surface regions. But I'm going to suggest something different. And this is work done in collaboration with Ed Silson and Annie Chan, both of whom are relatively recent faculty in the UK now. I'm going to suggest that actually the reason that you have this apparently redundant organization is to satisfy a retinotopic constraint. So if we look at the lateral surface of the macaque now, we can see that the lateral surface derives the majority of its input from the dorsal portions of the early visual field maps, which of course are representing the lower visual field. The ventral surface derives most of its input from the ventral portions of the map, so those that are representing the upper visual field. So my argument will be that the reason you have this dual structure is because you need to be able to represent these sorts of stimuli in different positions across the visual field. Okay, how do we test it? We can do that using PRF modeling. All we need to do is localize the areas. Here we have PPA on the ventral surface, its companion region TOS, or sometimes called OPA on the lateral surface. And then we can simply do the population receptive field study, and we find exactly what we predict, which is a bias towards the upper visual field in PPA and a bias towards the lower visual field in TOS. Okay, that goes right along with the predictions from the neuroanatomy, but it also kind of argues against the hierarchical interpretation, given that these two areas are in fact representing different portions of the visual field. Okay, it also opens up the possibility for another sort of strange behavioral prediction, right? In the PPA, we observed this strong distinction between open and closed scenes, but that distinction was much weaker in the TOS. So if TOS and PPA are actually representing scenes, right, but they're representing them in different portions of the visual field, it leads to a strange prediction, which is that in PPA, which does seem to care about this distinction between open and closed, your behavior should be better for making that judgment in the upper visual field than in the lower, because PPA represents this distinction and TOS did not. Okay, whereas for a dimension like man-made versus natural, right, we expect there to be no difference between the upper and lower visual field because it was equivalently non-represented. Okay, we can run a very simple behavioral experiment, present a scene very briefly, immediately mask it, ask people to make a judgment whether it's open or closed or man-made or natural, and we observe the expected difference. So here you've got accuracy difference between upper and lower visual field, and you see a significant advantage for the upper visual field for making open versus closed judgments, but not for man-made natural. Okay, so all of this is to say that we can take, by the application of sort of converging evidence from multiple domains in neuroscience, we can explain the structure of category selectivity, and we can make sets of interesting and somewhat novel behavioral predictions. But I want to now take a step sort of outside the domain of recognition, right, and move into working memory. And so this was work really spearheaded by Soo-Yeon Lee, now faculty in South Korea, and Chun-Wei Ting, a former graduate student of mine now in Brad Possel's lab. And this work was really motivated by a set of results coming out of a number of the labs showing that during the maintenance of information in working memory, you could decode that information from the multivariate response, but in posterior perceptual areas, which suggested that at least somewhat overlapping populations of cells were going to be responsible, right, for processing both incoming perceptual information, right, as well as the information that was being maintained in working memory. Okay, so if we think about the classic models of working memory, right, this is quite a change, right? So in the classic models, working memory is held as a sort of distinct process oftentimes proposed to live within a frontoparietal circuit that's responsible for maintaining information and then manipulating it. Effectively, information is then copied, right, from the perceptual areas into this frontoparietal circuit where it's going to be processed. Okay, that relationship, right, agrees to a degree with the existing neuropsychology data. Certainly when you have damage to the prefrontal cortex, you have severe deficits in working memory tasks. Okay, but our idea was that, in fact, where you maintain information is going to be based on the nature of the information that you are maintaining. And so we designed a task to look for this dissociation. So here, we're going to take a sort of standard working memory paradigm where you're going to be presented with two objects followed by a cue. And the cue is going to indicate which of these two objects you need to maintain during the delay. But then in half the runs, your task is either going to be to do a visual match. Did this fragment of an image, right, occur in the object that you were maintaining in memory? Or a non-visual task in which you are simply asked, is this item from the same category as the item that you were holding in memory? So effectively, therefore, during the delay, you either have to maintain visual information or abstract information. And our prediction is that you will find the information in posterior areas when it's visual, and in the prefrontal cortex when it's not. And that's exactly what we find. So we localize object selective cortex and then the lateral prefrontal cortex. And here you have the classification performance for decoding maintained information. And you can see that it's significantly above chance in PFS for visual information, but not non-visual. And then the nice crossover interaction that leads to the opposite result, right, in the lateral prefrontal. But now that we sort of have stronger evidence, right, that there is this co-localization of working memory maintenance and perception, then we can make a set of predictions, right? We should behaviorally be able to observe direct and bidirectional interference between these two processes. That interference is going to be obligatory, right? If this population of cells is currently maintaining some bit of information, then incoming perceptual information will interact with it. And that interaction should be consistent across delays. And then moreover, the strength of the interference is going to be predictable from the known tuning curve properties in these perceptual areas. And so in order to test this, I think we'll skip a little bit because I'm running out of time. So in order to test this, what we're going to do is lean into the things that we already know about, in this case, orientation and color selectivity. We have some understanding of the underlying tuning. And the first thing we're going to do is try to demonstrate that the influence of incoming perceptual information on working memory is going to be commensurate with those tuning curves. Okay, to do this, we run a relatively simple experiment. You're presented with, in this one, a colored disk. You simply have to maintain that color in memory. Following that encoding period, participants do an RSVP task where they have to press a button anytime a white letter appears during a stream of letters. Behind that letter, and completely irrelevant to the task, is another colored disk, right? And what we're going to do is systematically vary the similarity between the item you're holding in memory and this distracting disk, which is completely task irrelevant. Following this, participants then do a continuous memory report where they simply click on a color wheel to indicate which color. And what we're going to do is to now measure the amount of bias which has been introduced, right, into the working memory report by this irrelevant distracting stimulus. And what we're going to anticipate is that we should see a recapitulation of the tuning curves, right? The more similar these two things are to each other, the more overlap in the population of cells that the perceptual stimulus and the working memory stimulus are going to be driving, and therefore the more of an interference effects we should observe, and that's exactly what we get. We can play this game for color. We can also play the game for orientation. We run exactly the same experiment, but now with Gabor that has an orientation to it, and we get exactly the same result. The more similar the perceptual item is to the working memory item, the more it biases that working memory report. Okay, but now we want to show the interference in the other direction. So we want to show that what you're holding in mind actually changes what you are perceiving. And in order to do that, we have to engage in a somewhat more complicated experiment. So I apologize in advance. So what we want to do is first pick a task that's really a low-level perceptual task, because we want to get a pretty clean measure of that. And so we settled on deriving discrimination thresholds. And so the task is going to go as follows. Participants are going to be presented with a colored gabor, and they are going to be instructed to maintain either its color or orientation. Following that, they're going to do a color orientation discrimination while they are maintaining that information in memory. And during that discrimination, we're going to derive their discrimination threshold. Following the discrimination, they then do the continuous working memory report for either color or orientation. The first thing we're going to manipulate is during the discrimination task, we're going to manipulate, obviously, the similarity between the two gabors. And so that's going to allow us to derive these standard psychometric curves, and from them we can get out a threshold, at which point they are saying 65% of the time that the two stimuli are actually different. Okay. We're also going to manipulate the match between the item, the feature rather, that's being held in memory, and the feature that forms the basis of the discrimination. Right. So in some cases, participants will be doing color memory and then color discrimination. That's one group of people. Another group of participants does orientation for both tasks, right? But then two other groups of participants are going to be doing the non-match conditions, where the items that they're holding in memory, the feature they're holding in memory rather, doesn't match the feature that they're doing the discrimination on, right? And so even though the trial sequences are actually identical for all four groups of subjects, the only thing that varies is the task instruction, we nonetheless anticipate that we should only observe this interference effect during the matching conditions, right? Because that's where you're actively maintaining and driving those overlapping populations of cells. Okay, now, finally, what we're going to also manipulate is the relationship between the two discrimination stimuli and the item you're holding in memory. So let's take a simple condition, right? So in here, these vertical black bars are going to represent the discrimination stimuli along the sort of color spectrum. Now let's imagine to begin that the item you're holding in memory, you're holding a color in memory, and it lies between these two discrimination stimuli. What we think is going to happen, and what we kind of know from our previous experiment, is that the memory stimuli is effectively going to pull the population response towards itself, right? You've already pre-activated some population of cells that represents this middle color, and now the population response for all three of these colors is going to get intermixed. Effectively, that's going to draw the perception of the discrimination colors, right, towards this color that's in the middle, and it's going to draw them both in equally because it's equidistant from them. Okay, effectively that means that the perceived distance is going to be smaller than the actual distance, and therefore your discrimination thresholds should be higher, right? You're going to need more of a difference between the two stimuli to consistently say that they are actually different. But now contrast that to a case where you're holding an item in mind that's to one side of these discrimination stimuli. In this case, it's still going to draw the population response towards itself, but that effect is going to be larger for the stimulus that's closer to it than the stimulus that's further away. Because of that, the perceived distance between the two is actually going to be greater, which should lead to lower discrimination thresholds. And so our fundamental prediction is you will have lower discrimination thresholds inside than middle during those match conditions. And of course, this is exactly what we find. So here's the psychometric curves in the raw, right? What we can do is go ahead and then model them using a sort of standard psychometric function. And from that, we can derive the thresholds. Here you can see that the threshold for the middle condition for color memory, when you're doing color discrimination, right, is shifted, right, relative to the side condition in the predicted direction. That shift is absent in the equivalent non-match condition. We can do the same thing now for orientation discrimination. We see the shift during the match condition, and the shift is much smaller and in fact not statistically significant in the non-match, right? We can summarize those in a nice crossover interaction plot, which shows that what you are holding in mind, it does affect what you see, right, when you have a match, right? When you are holding in mind a thing that directly relates to the incoming perceptual information and the task that you're trying to do with it, then in fact you do get this bidirectional interference, exactly as is predicted from the co-localization of these two different processes. And so in this last section, which remarkably we may have time to actually do, we're going to talk about some more domain general mechanisms, right? So what we've been talking about up until now is really specific patterns in the neuroanatomical connectivity, or specific results coming from the fMRI literature showing co-localization. But there are also a number of things that we know about just the ways that neurons behave, right? How they actually respond, and how their synapses, for example, change over time. That do make some interesting behavioral predictions, sort of regardless of the exact details of where things are being implemented or not. And so where I've done the majority of this work is looking into attention and learning. And this is work that I actually did, some of which during my sort of graduate career, and then more recently in collaboration with Steve Mitroff. So let's begin with visual attention, right? If we look at the brain as an integrated circuit, sort of regardless of exactly which areas are going to be driven by any given task, and those areas will vary, right? Even between tasks that are on the surface somewhat similar. But regardless of exactly which network of regions is active, because everything is an integrated circuit, then the consequences, right, a change in the response of one circuit is going to have consequences, right, for all of the other circuits to which it's also connected. Okay, this in fact is one way of interpreting classic attentional effects like object-based attention, right? So if we take the really sort of 1950s broadband models of attention, then we think of attention as largely a spatial phenomenon, right? Where we can cue a location in space, and what's going to matter in terms of how quickly you're going to be able to detect some other stimulus that's unexpected is purely going to be distance from that cued spot. And so in a display like this, where you have a fixation cross on a single object, if I cue one side of the object, then these targets, right, which are exterior to the object, are equidistant from the cue, and also equidistant from fixation, so we expect their behavior to be equivalent, right? That's if attention was purely spatial. But in fact, we know that attention, right, also cares about objects. And we know, well, actually, this was one of the first studies to directly demonstrate it, but the consequence of object-based attention extends not just to that object, right, but also to the spatial locations within that object, right? In other words, a facilitation is going to occur, right, for other locations within an object that can contain targets, right? The consequence of that facilitation is going to be an interaction, right, where now what we might predict is that, in fact, it's no longer distance just from the cued location, but some weighted average distance from the object that matters. And because of the positioning of these two targets, this target is always going to be closer on average to the object than this one, allowing us to predict a difference. But we can take it a step further, because we also know that it's possible to cue features, right? Things like the concept of red, right? The color red can be cued. Let's propose that when we cue an object, we're not just cueing that location or that object, we're also cueing the high-level description of that object, in this case, say, rectangles, right? If that's the case, then if we add a second object into the display that's identical to the cued one, it should also get an attentional benefit. That will also have consequences for the spatial representation of this object, which will in effect mean that now it's average distance from both objects that matters. And once again, we're moving towards a point of equivalence between these two targets. On the other hand, if we have a second object that doesn't match, then we can expect less of a spread of that facilitation and more of a difference between these two exterior targets, right? In addition, we can also predict that the cost of shifting your attention from one object to the other should be lower in the condition where the two objects match than when they don't. And to make a rather long-winded story short, that is exactly what we see, right? We can play this game for shape, we can play it for color, and no matter what, we get essentially the same result, which is a larger difference, right, between the exterior targets and the non-identical than in the identical case, and a larger between object cost as well. Okay, this though holds really regardless of which dimension is being cued. Right, so we can even demonstrate that it holds true for high-level abstract category, right? So here we have two different displays, one of which looks like an H and a lowercase h, and the other of which looks like an uppercase H and a 4. In fact, the 4 and the lowercase h are exactly the same object, just rotated at 180 degrees. And nonetheless, the exact same effect plays out, right? When you interpret that second object as a lowercase h, it accrues more of an attentional benefit, right, when you cue the h than when you interpret it as being a 4. Okay, and all this is to just say that once we start thinking about the brain as this integrated circuit, we can begin to think about some of these processes like attention as also distributed processes, which are going to play out in the dynamics occurring between even separate populations of cells, right? All of which have some common underlying factor that keeps them connected. In this case, space, right? The object occurs at a location in space, as does the features, right? As does the abstract object category as well. And so long as there is some basis for that communication to be occurring, we're going to observe it, and it should bleed through into the behavior. Okay. But we can also take this even sort of a little half step further, right? So let's think about now not just the fact that the brain is an integrated circuit, but that a lot of the changes which are occurring in that circuit, at least in the slice physiology, appear to be local. Right, which is to say neurons are relatively simple little creatures. They do not know to what grand process they are contributing. What they do is to remodel their synaptic connections on the basis of some combination of feedback signals and Hebbian learning, right, which is the domain of computational neuroscience to sort of tease apart. But ultimately, a lot of those changes are going to be local to those individual synapses, which means that there should be a continuous aggregation, right, of statistical evidence, which is always occurring, right? And that aggregation is going to be more or less, have more or less of a profound effect on the behavior, depending on, how relevant the particular bit of information is for the behavior that you're measuring. But regardless of what the feature is, even if it's task irrelevant, what we should be able to observe is a continuous aggregation of information. And moreover, that aggregation should be domain general. Right, the same basic learning rule is most likely going to apply, right, regardless of which domain we happen to be studying. But of course, this is a really difficult thing to get at because most of the time our experiments are well designed and they are not meant to study this sort of a phenomenon, and so everything is balanced, which means that by the time you're out to trial 30 or 40, there really is no evidence in favor of one feature or another, or one kind of trial or another. And so in order to study this, you really need to have a very unique sort of a data set where you've got a large number of people that have all experienced a very diverse set of early trials. And as it turns out, Steve Mitroff, through really pure grit, managed to acquire one. And so the data I'm going to present here in this last bit is gleaned from Airport Scanner, which is this smartphone game where you take on the role of a security screener working for the TSA looking for dangerous items in bags. And to date, over 15 million people have, for some reason, downloaded and played this game and have performed what amounts to 3.7 billion trials of visual search. Okay, I don't know why they do this for fun. They're not getting paid. In fact, in some cases, they pay the company that put out the game to do this, but we're going to benefit from it anyway. The other nice thing is that the game also has, in addition to this visual search component, it has a couple of other mini games within it, which are also sort of accidentally recapitulations of standard cognitive psychology tasks. So we're going to be able to look for changes which are going to be occurring across tasks and see if there is, in fact, any sort of consistency. Apologies for the axes here, which seem to have shifted a bit. So the first thing I'm going to show you is data from this object sorting task. The object sorting task is really very simple. A single object appears on the screen, and participants simply have to swipe it up or down, depending on whether or not that object is allowed to go on a plane or whether it's prohibited. And so the first thing that we're going to do is simply take advantage of the sheer volume of data we have, and in a totally model-free way, simply try to quantify performance as a function of trial number and the proportion of prior trials in which a prohibited item was shown, right? And so what you have here on the y-axis is going to be the accuracy on prohibited trials. And then on the x-axis is which trial number is the data is going to be gathered from. And then the different colored lines are going to be indicative of how many of the prior trials contained a prohibited item or not. And what we'll see as this sort of plot fills in, right, is that the more prohibited items occurred in the past, right, in the preceding trials, the more accurate participants are at correctly classifying an object as being prohibited, right? And we can see this play out in two different ways. So one, we can see the difference between the lines, right? And that is directly the number of previous trials that were prohibited or not, right? We can also see that the more trials that you have done, right, even if all of them were prohibited, performance is better for 6 out of 6 than it is for 5 out of 5. Okay, there's a very easy way of aggregating all of that together, which is to just use a standard metric of statistical evidence. In this case, the binomial z. You can also use Bayes factors, chi-squareds. All of them produce numerically similar results. Here, the binomial z, what we're going to do is simply take all of this data and now plot it as a function of the binomial z-score rather than as trial number and number of prior prohibited items. Okay, and we can do this one-to-one mapping and then transfer them all over, and we explain that entire kernel, right? So in fact, your efficiency at least over this early set of the first 10 trials, your efficiency at accurately classifying a thing as prohibited is directly related to the statistical evidence from the previous trials that you're going to see a prohibited item. Okay. What we can do then is work our way now into the more complex data. So we can go into the visual search. And the visual search data is going to help us to deconfound exactly what's driving that learning effect. In the object sorting task, you know, the trial condition and the nature of the object that's being presented and even the response are all related. Whereas in the visual search, the responses differ. There are features, as we'll see, of the stimuli that are largely task irrelevant, which means that we can test and see whether or not a much more diverse array of features are also showing the same basic learning. To sort of shortcut showing you all the intermediate steps, we'll skip directly to the binomial z-score plots. Here I'm going to show you hit rate. So this is the proportion of times that participants accurately classify and tap on an item in the bag as being dangerous. And you can see that your hit rate is again now directly related to the evidence for now previous target present trials. The more target present trials, the stronger the evidence for targets being present in bags, the more accurate participants were at detecting those. They were also faster, right? They were also more quick to find those dangerous items. What we can also do is, like, this effect also holds for target absent, but in the opposite direction. What we want to do now, though, is move away from these features that are directly related to the response and instead deal now with features which are orthogonal, right, to whether or not a target is present. And like all good visual search researchers, we go ahead and look at the distractors, because a distractor can be present regardless of whether or not a target is. And so what we're really looking at here is how quickly, right, or how do participants learn, right, in order to deal with these distractors, even though none of this has a direct influence on the actual response that they're making. And we can run this for a wide variety of different features. We can look at color. We can look at the presence of particular distractors here. This plot is showing the presence of headphones for both present and absent bags, or the absence of those. We can look at the type of bag. It really doesn't matter what we look at. All of it comes out the same. Regardless of the exact feature that you're looking at, regardless of whether or not it is directly task relevant, you see the same basic learning curve, so that over time, the more, in essence, evidence or opportunities you've had to deal with now this utterly irrelevant, distracting information, the more efficient participants are at both target present and target absent trials that contain that item. Okay, and that's what's being plotted here now for hit rate on the top and for hit time on the bottom. The relationships are not quite as strong as the ones that we observe for in the object sorting task, or even for the trial type, right, target present or absent in the visual search, but they're nonetheless there. Okay, and that's consistent with this idea that you know, in addition to all the cognitive effects, which are going to be emphasizing or de-emphasizing particular features and therefore driving learning to be stronger or more present, right, for some features than for others, underneath all of it, there is still this sort of task relevance invariant, right, learning, which is occurring. and is most likely going to be driven by some sort of a local synaptic mechanism that's relatively sort of transparent to exactly what the task demands are, right? The simplest account actually for this sort of a learning kernel is really just Hebbian learning, okay? There are a couple of implications for this that we are currently exploring. What we're doing now is looking through old sets of laboratory behavioral data to see whether or not the same kernel, right, can be found in those. And the short answer to that is yes. In fact, you can see it. And then the question is there something to be gained, right, by partialing this out? If we are trying to study this cognitive effects, right, which we think are, perhaps divorced from these sort of local changes, then it's possible that this is essentially an uninteresting and maybe even countervailing source of noise. And that by partialing this out, we'll get an advantage, the same way that we do when we partial out, for example, motion parameters from our fMRI data. The other thing that we have to grapple with is that we may not be behaviorists, but certainly some aspects of the physiology are, which is to say that they're constantly accumulating information about the context, right, that the participant is in. That is partially, right, one of the reasons why behavior does vary so much from paradigm to paradigm, person to person, task to task, because it's picking up on that context, but it's also going to be an aspect of the physiology that we need to understand, right? That change in behavior must be supported by a change in the underlying physiology, and right now all of that is kind of mixed together. Right, so if we're trying to understand the physiology of our cognitive effect that we, hope and pray is somewhat more stationary than this, then there may also be an advantage to partialing this out, right, and studying it distinctly when we look at the physiological response. Okay, and so I'm only a few minutes over, which is great. So I'm just going to very quickly summarize and just say that, the enterprise of cognitive neuroscience is really extremely difficult. On the one side, we have a bunch of physiological data, the complexity of which is simply staggering. On the other side, we have really good quantifications of behavior, but the mapping from one to the other requires us to jump through, in essence, infinite space of nonlinear models, right? And those models are, moreover, going to be changing, right, from person to person at some level of fidelity, and then also potentially from trial to trial as the model itself changes in order to become more efficient and adaptive at our tasks. And so to my mind, the only way that we're going to make, well, not the only way, one of the ways that we can try to surmount this gap, right, is to try to make these sorts of predictions, right? Try to tie aspects of the behavior directly to the physiology. And then eventually use that in order to refine our understandings of the underlying cognitive mechanisms. So I'm going to stop there, but thanks everybody for your attention, and I'm happy to take any questions.
Thanks so much, Kravitz. So yeah, I'd like to open the floor for any questions that people might have. So maybe we can start with students.
So I have a question. I think this is a really cool talk. And my first question is, if the hypothesis is those category selective region is driven by the underneath neural circuitry, and that's those based on the neurophysiology data can predict the response. So to what extent this prediction can also predict the system where there are lesions? Does that make sense?
Yeah, so this is an interesting topic, actually, which weirdly there's a sort of nascent literature in the macaque on. So one thing that you can do, for example, in the macaque that you can't really do in the human is you can apply a pretty clean lesion. And so you can do things like lesion monkeys at different points in development in order to see, when you interfere with ongoing perception and when you don't. And so one of the interesting things, right, is that early on in development, if you sort of cut, right, so let's say that you apply a massive lesion that blows TEOD to pieces, right, then you really do get long-lasting behavioral problems, right? So the monkeys will eventually recover, but early on at least, right, it's very difficult to get TE to actually respond to anything anymore, because the primary input into TE is from TEO. On the other hand, as you go to more adult monkeys, when you apply those lesions, it seems as though this indirect pathway, right, has sort of learned enough to be able to drive TE response. And so there is this bizarreness to the underlying lesion data, right, where depending on where you are in the course of development, right, the neuroanatomy actually helps you to explain the differential patterns of deficit that you observe. One other kind of prediction that you can make in the human data that I don't think has been tested exactly, though maybe in the language literature is vast and I'm giving it short shrift. To the degree that you find sort of overlap in what parts of the brain are driven by processes, then that overlap should then help you to predict exactly the kind of comorbidities that you should observe in lesion patients, right? So it's very rare, and in fact, I would say vanishingly rare, to find a pure lesion case. But some amount of the neuroanatomy can help you to make a prediction about the impurities, right, rather than just documenting them.
Thanks.
Sure.
I have a question about the app data set that you were talking about at the end. Is there any way to know what the ages of those participants are? Because I would expect there'd be a lot of like children in that data set.
So what Steve Mitroff did do, was there is a version. So he contacted the company that made the game, and they were just, they had just made a game, and they started logging data. And then eventually they stood up a subsection of the game that was a bit more controlled, a bit more like a standard psych experiment. And then as part of that, participants then had the option of filling in a survey that gave us some of the demographic information. In practice, what that demographic information shows is that the age ranges from about 14 to 20, with most of it being in that sort of like 17 to 19 range. That's the only data that I have that would give us exactly ages. Other than that, what we have is a lot of data about sort of time zone and what times people are playing.
Hi, Dwight. Thanks for the very comprehensive talk. I have a question about the second part of your talk.
Sure.
I'm curious, did you observe a simultaneous attractive bias for the perceptual representation towards the visual working memory and also vice versa? And also,
yes.
do you predict that there would be more or less bias for a working memory versus a perceptual representation?
Yeah, so I think it's always going to be difficult to sort of titrate and balance those two measures against each other, just because the perceptual task and the working memory task are happening at different time scales. So putting that aside, my prediction to your last question would be that the working memory representation is less stable, right? So the one bit of evidence we do have about that is when you make the delays longer, what you get is a stronger effect of perception on working memory and a weaker effect of working memory on perception. So over time, there's some sort of a decay in the working memory representation that attenuates, right, its contribution to that interaction. In answer to the first part of your question, I think yes, these two experiments were exactly designed to show that you do get a bias in both directions, right? Where in the first experiment, we show that the perceptual information creates an attractive bias, right, in the working memory report. And in this one, what we're really banking on is that the working memory is creating an attractive bias in the perceptual representation. And so I think that the bidirectionality is there. Figuring out exactly the relative strengths is going to be challenging because the two tasks are so different.
Thanks.
Sure.
I have a few questions, but so it's a great talk. Thanks so much. So a bunch of the work that I do involves testing the connectivity hypothesis as well, and looking at the development of functional organization, how that might be driven by connectivity. So I buy, your underlying hypothesis here. So I'm wondering if you have looked at similar sort of macaque work-driven neuroanatomy and how that might give us a distinction between the different face patches. So like OFA, FFA, or the two different patches of FFA, as well as STS. And I know that the monkey versions of them are quite different, right? They're more superior. It's it's not an inferior temporal cortex. So I'm wondering if you've looked at that specifically.
So let's see. There's a lot to unpack actually in the answer to this. So to begin, the first order answer to your question is that Salim and Doris Sau, I think, have an ongoing collaboration looking at exactly this issue. Whether or not there is enough detailed tracer work that's been done in the macaque to differentiate the connections between the face patches is tricky, because the patches in the monkey are not quite as stereotypical as the ones that you see in the human. And so I think to do that work well, you need to do the imaging guided tracer work, which is, I think, where that collaboration is sort of heading. So the short answer to your question is I don't have the answer, but I think people are working on it. The other interesting aspect about the macaque is, if you go back to the old recordings that like Dave Parrott was doing and Ed Rolls back in the 80s, they were actually recording face cells in the inferior temporal cortex on the inferior surface. The problem is that when you are constrained by the monkey imaging, the size of the jawbone and the nature of the monkey imaging, the muscles around the jaw rather, make it really difficult to image down there. And so it's not totally clear yet what the homology is for the face patches, like which one of them is FFA, which is OFA. Can we even think about it in that way? All of that remains really unclear. I think the future is going to clear a lot of this up. I think that like Marge Livingstone has some projects looking at deprivation in young monkeys and looking at the consequences for functional organization, and the collaboration between Doris and Salim, I think, will eventually produce firmer answers. But for right now, I think a lot is up in the air because we know there are face cells on the inferior surface, but we can't seem to cleanly get an imaging signal out of them. So it's just tricky.
Okay, thanks. And one short question. So this is about the PFS activation during working memory. I'm wondering if you modeled the delay period when there wasn't a visual image on the screen, and whether or not you saw responses in object selective cortex during that delay period.
I see. This is an interesting question. So the pure activation measures, right? Just was there activity in PFS or not above baseline, really didn't show very much. I think they were active, right? But there wasn't any difference between the two kinds of runs, right? So whatever that activity was, leaving aside the information metrics for the moment, right? It's difficult to interpret it. Is it leftover activity from the object presentation? Is it something else? It didn't differentiate between the stimuli. It didn't differentiate between the conditions. What did was the multivariate information, and that's sort of consistent with that literature. I think that even if you go back to some of the classic working memory studies, they ran into the same problem. So if you look at like Susan Courtney's stuff from the 90s, all of that posterior activation is there, but there's no way to interpret it because the paradigm kind of, you end up doing the encoding and then no one leaves a gap long enough for the bold signal to completely decay. And so you kind of have to go to paradigms more like this one, and then the information measures, I think, to really look at it. That being said, it's at some smaller level, right? At a level beyond the fidelity of the studies, there probably is an overall activation difference. We just can't get to it in a reliable way.
I do, just to jump in there, I do recall from some of Susan Courtney's work that at the really longest delays, you started seeing some breakdown in infratemporal type processing. And then she argued that there was a shift in frontal maintenance that went from more visual to more analytical. But again, I totally agree with your point that that kind of method was really hard to get at that question.
It is, and I think though that you're touching on a really interesting point, right? Which is here at least, right, this shift between visual and abstract information, we're setting it up and at the level of the task, and it's done by run. So the timing here isn't really all that informative, but it does seem like it, it occurs within this sort of a delay period. But it is an interesting question. What are the limits of the maintenance of information in the posterior cortices? Because at some level, their natural state has to be decay, and kind of profound decay, right? If you're not actively maintaining it, then you want it gone, because you're going to move your eyes and you're going to move your head and you're going to move to a new environment. And so there's definitely going to be some sort of a transition, right, where some poorer version or analytical or abstract version of the information is all that you're able to sort of hold on to, right? In a biological system, you're not going to be able to do this perfectly forever. We have run experiments out to about 8 seconds in the behavior, and the interference effects are consistent through that range. But beyond that, right, into the 30 seconds, 1 minute range, I don't know where the limit of the system is going to be, but there almost certainly should be one.
Any other questions? All right, well, thanks again. This was a wonderful talk. And thanks again for coming to OSU virtual. So if anyone has additional questions for Dr. Kravitz, feel free to email him. And have a great Friday, everyone.
Absolutely. Thanks for inviting me, and thanks for the attention.